central dogma is a theory that was given by Crick. He stated that a DNA forms an RNA through transcription and an RNA forms a protein through translation. When DNA forms another DNA, it is called as DNA replication. And we are going to discuss DNA replication in this video. DNA replication is known as a semi-conservative mode of replication. Now, why do we call it a semi-conservative mode of replication? So, DNA is this helical structure, but replication happens by separating the two helically wound strands apart, and each of the strand will form one new strand. The new strand will then helically wind along with one of the previous strands or one of the old strands which acted as a template for the new strand to be formed. Hence, a new piece of DNA will include one new strand and one old strand. Hence, DNA replication is a semi-conservative mode of replication. The first step in this procedure is a molecule called DNA finding a region on the DNA which has a lot of AT pairings. This molecule is going to initiate the replication by initiating the breakage of these AT bonds. Why is this molecule only looking for a region which has a lot of AT pairings? Why not GC? So if you know, A and T bond with 2 hydrogen bonds, while G and C bond with 3 hydrogen bonds. It's easier to break A and T with just 2 hydrogen bonds, and it will require a lot more energy to break G and C, which have 3 hydrogen bonds between them. Hence, DNA A molecule is a very smart molecule. It's come up with a strategy that it has to find a region on the DNA which is heavily having AT pairings so that it can initiate the replication on this point. This part of the DNA which has a lot of AT pairings is known as the origin of replication, because this is where replication will originate. DNA A molecule is just going to initiate the breakage of A and T bonds. Then helicase, which is an enzyme, will start breaking or splitting apart the 2 strands while opening this small section of DNA which is going to form the replication bubble. The parts that precede and succeed this replication bubble is bound to get supercoiled. In order to maintain the structure of the DNA which precede and succeed this replication bubble, we have an enzyme called Topoisomerase. It prevents supercoiling of the other sections of the DNA while the replication bubble is being formed. DNA or RNA often form this bubble when there are no complementary base pairs. But in this condition, we have complementary base pairs, but this bubble is being formed. That's because Helicase enzyme is opening up these 2 strands of DNA. But due to their complement pairs, they are bound to stick together and form their hydrogen bonds once again. In order to prevent this from happening, we place some molecules like a barricade, which are known as single strand binding proteins. They keep these 2 strands apart and help maintain the replication bubble. Next, we have the hero of the story who's arriving, DNA Polymerase III. This is an enzyme which performs the function of DNA replication. It's a highly efficient enzyme which has an amazing speed, and it also performs the function of proofreading what it's done. But since it's so efficient and it's so profound in its work, it has a few demands which needs to be fulfilled in order for it to start its work. Some of the demands of this enzyme is that it's going to form the new strand from the 5' end to the 3' end. The second important factor that it requires is that it needs a small double-stranded portion in order to initiate its DNA replication. Giving it this double-stranded portion seems a little bit difficult because we've just used special enzymes which are helicase enzymes to split open this part of DNA which we want it to replicate. So here we call another enzyme who comes and aids us help. This is the RNA Primase. RNA Primase is going to select a few sequences from this opened up structure or the replication bubble, and it's going to make an RNA which is complementary to a few sequences of this replication bubble. This small structure of RNA which is complementary to a few base pairs on the DNA is known as a primer. This primer is going to attach on that small patch of DNA, providing the DNA polymerase a double-stranded structure for it to initiate its polymerization. Now this DNA polymerase, which is DNA Polymerase III, is a DNA-dependent DNA polymerase. Why do we call it DNA-dependent? We call it DNA-dependent because the template strand which it uses to read the code is a DNA itself. And since it's forming a DNA, it is a DNA polymerase enzyme. Hence, this enzyme, which is DNA Polymerase III, is a DNA-dependent DNA polymerase. Now, if we look at both the strands, the DNA polymerase enzyme is going to start replicating and making the new strand of DNA from the 3' end of the template strand to the 5' end of the template strand. Once it has completed the replication of the replication bubble, helicase is going to arrive once again, and it's going to cut through and split open the parts that follow the replication bubble. And the strand which is on top, which begins from the 3' end and moves towards the 5' end, since DNA polymerase has this double-stranded structure, so it does not need a primer once again. So it's going to continue its replication as and when helicase keeps opening sections of the DNA that follow the replication bubble. But if you notice the strand at the bottom, which begins at the 5' end and moves towards the 3' end, as and when helicase cuts open or splits open the two strands of DNA, we do not have a double-stranded portion for the DNA polymerase to come and start polymerizing the DNA. Hence, we call RNA primase once again. RNA primase molecule is going to form the small structure of RNA or the small stretch of RNA which is complementary to these few base pairs on the DNA, and this is going to provide this double-stranded structure for DNA polymerase to come and to perform DNA polymerization or DNA replication. This process continues for the strand at the bottom. If you notice, the strand on top has this continuous formation of DNA as and when helicase opens a part of the DNA. Hence, the strand on top is known as the continuous strand, while the strand on the bottom has DNA formation in fragments. Therefore, the strand at the bottom is known as the discontinuous strand. The fragments on the strand at the bottom are known as Okazaki fragments. Towards the end of a DNA, we have the stretch of DNA which does not code for anything. This is known as telomere. Telomere is simply a random collection of few codes which do not mean anything and are an indication for the DNA polymerase to jump off from the DNA and to stop the replication process. After every replication, the telomere shortens. That's because DNA polymerase stops coding before the telomere ends. Ultimately, the telomere is lost and the DNA can no longer replicate. Therefore, the DNA has a limited amount of divisions that is possible, and this limit is known as the Hayflick's limit. Once replication is done, both the leading and the lagging strand will have primers. The leading strand will have just one RNA primer at the beginning, but the lagging strand has several Okazaki fragments which have one primer each. Hence, the lagging strand has several primers and the leading strand will have one primer. Since this is DNA replication, we do not require an RNA here. Hence, we have the enzyme DNA Polymerase I, which is going to extract or remove these primers from the replicated DNA. Once the primer is removed from the continuous strand is all ready and it's completed. But once primers are removed from the lagging strand, these Okazaki fragments are still fragmented. In order to join all of these fragments, we have an enzyme called DNA ligase, which performs the function of ligating or joining all of these Okazaki fragments with one another. Once DNA ligase joins all of these Okazaki fragments, the lagging strand has also completed its DNA replication. So this is how DNA replication occurs. I hope this video was helpful in understanding this concept. That's all we have for today. Thank you.